Okay, so we're gonna go over the nucleic acid lab. Okay, so really quick, just to kind of hit up on the protonation for the amino acids, what that means is how do that's why I kind of gave you examples for the amino acids that we covered on our last lab that we did have. I talked about our acidic amino acids and our basic amino acids, and to kind of talk about how those affect the body. It's the first one. There are many files in this lab. Yeah, the first one. The thing is I just want to talk about what he's looking for as far as how it affects the body. We want to maintain a physiological pH, right? And that's like 7.4. So if we have these amino acids and they're acidic or they're basic, which means they have a negative or a positive charge, or they're in this zwitterionic state, if we have all of these charged amino acids, okay, in our body, how is it that we can maintain this physiological or homeostatic pH? Okay, so what that means is those interactions between those amino acids, their interactions are what help keep us at a physiological pH. So far as that question goes, and I'll just relook at it, I'll look at it again so that that way I'm answering it 100% for you. It says, draw the full structure for the amino acids and showing the protonation state as they exist. So and then comment briefly on how that side chain influences the properties of your chosen amino acid. So the first part, that protonation state is, like I said, is the positive or negative. So there's no elaboration on that. You can literally just say, yes, answer it for every amino acid, because you should have done 4 amino acids, right? Or 5 amino acids. And so if you just stuck to the acids and the basic ones, you can point out and state the same for all of them, the carboxylic or the amine, right? So those, because they have the amine group or because they have the carboxylic group, it is because of those that they are either acidic or basic, and that side chain affects how it functions interacts with other amino acids. Okay, so for instance, lysine and glutamate, right? We have an N group, an amine, and then we have a carboxylic. Okay, and this is kind of what's going to lead us into our nucleic acids, right? Because a nucleic acid is what? What is the basis of nucleic acid? It makes up a nucleotide, right? So if we're talking about Watson and Crick and nucleic acids, lysine and glutamate, they actually will form, and I guess we should talk about salt bridges. They'll form a salt bridge because we have lysine that's positive with an amine, and then glutamate with a carboxylic acid, which is a negative. So we have a positive and a negative. And remember, carboxylic acid, the structure for carboxylic acid, we already know, is this, right? And there could be an OH here, but nonetheless, we have this from lysine, some amine group. And so what do we have here is we have an amine, we have a lysine that's a positive charged N, and this is overall negatively charged N, okay? So individually, these amino acids are positive and negative, separate. So that is how they live in the protonated state in the body. And then they come together, this is their interaction, they would come together. And not only are they experiencing, number one, hydrogen bonding, answering number one on the amino acids, yes. Number one on the amino acids lab. So not only do they exist, not only will they hydrogen bond from this H of the amine and this available O on this carboxylic, but you're also having some sort of ionic interaction. And this is the definition of a salt bridge. Okay, and I know that Tom will put that on an exam. No, you're good. You're good, Caitlin. So that is the definition of a salt bridge. It's the interaction that consists between these covalent interact covalent amino acids, and it has to be with through an H bond, and they have to be ionic. Remember, ionic is just what their charge states are. And we've already determined that these terminal ends of these amino acids are positive and a negative, right? So this would be like, this is glutamate, and then this is lysine, okay? So that's why I had you focus on the acidic and basic amino acids, because there's 20 of them, okay? But if you know that at least those 5 that are basic or acidic, then you'll be able to, and know their structures and keep practicing them, you'll be able to notice if that the other ones are all nonpolar covalent, okay? Because they're not charged. Know the charged ones, they're less than the ones other than memorizing 20 amino acids. Okay, so if he talks about phenylalanine or methionine or other ones that are not from the 5 that I've given you, you can automatically just say those are nonpolar and they're not charged. Okay, because he will do that. So this is how you'll answer number 1. You can go into the salt bridge and say that's what will happen, but just to reiterate, I would draw those structures for those basic and acidic amino acids. And that's how they exist, with their carboxylic and their amine groups, okay? And then it says to comment briefly on how the side chains, because they can interact and form potential salt bridges through their positive and negative of other amino acids, they can form salt bridges. Their charges help indicate where things will start to connect, okay? Any other questions on the amino acids? Does that kind of help clear things? Hopefully it does.
Yes, okay. So their salt bridges will help determine where the aminos will interact. Is that correct?
Yes, those salt bridges are indicative of where those amino acids connected and interacted. So if you see a salt bridge, and you will be given structures that do form salt bridges, you know that a salt bridge has to take place between some sort of hydrogen bond and an ionic bond. So you have to have an ionic interaction. So if you were given multiple structures, and he's done that before on exams, that you can be able to look through, for instance, if you're given something that looks like, before we begin the lab, I'll just show you. If you have something that looks like this, okay. Versus something that looks like this. You need to identify this carboxylic acid. So if anything were going to form a salt bridge, he could ask you which one of these structures is most likely to form a salt bridge. This is not going to form a salt bridge. First of all, we've got this cyclic, this aromatic. We do have this OH, right? So it's going to throw people off because I told you about hydrogen bonding, and this H could leave and come back, right? But you have to look for a carboxylic acid. And there'll be a lot of other, he might even throw in a structure that looks like this. And people will get thrown off. And the explanation for arginine would be the same as lysine because, yep, yes. So, students will get thrown off by this because we know that we see a lot of this. Okay, carboxylic and amines. This is the N term, this is the C term, and people will see this NH2, but is this NH2 positive? And if it were positive, it's just because you have a positive here and you have a negative here, then this is a Zwitter ion, okay? So this is gonna self-interact to maintain a neutral overall net charge. This is not going to form a salt bridge. Why would I form a salt bridge between 2 substituents on a carbon? Okay, a salt bridge takes place between 2 different amino acids. And the question could state, which one is most likely to form a salt bridge. This will not form a salt bridge, okay? Just because it has this amine part, this amine complex, and this O minus, this is not a carboxylic acid, and this would, I would just denote it as the amine terminus, okay? Or closer to an amine terminus. You need to be able to identify that the structure with a carboxylic acid would be most likely, because this thing is ready to go. It's ready to go look for something that's positive, another positive amino acid, okay? So this is the key identifier when trying to understand how salt bridges are identified. Okay, let's go ahead and start with nucleic acids. Let me just get this sheet out. Okay, the first says you have to read all these things, blah, blah, blah. Okay, no, we're not gonna do that. Well, I'm not gonna do that, you guys can do that. So number one, it says, please draw two Watson and Crick base pairs and show how GC and AT recognize each other through or by hydrogen bonding. Okay, that's very easy. So you basically can, I'm gonna draw it actually from a chemical standpoint, because from a structural standpoint, because that's how biochem works. We draw structures for everything. And it says just to draw the base pairs. So I don't really know how specific he wants to look at, but you could at least impress your TAs that aren't me with drawing these. So let's draw, we know what base pairs go together. We have these, we have all of these. These are our base pairs, right? Well, these are our bases. And which ones go together?
A and T and C and G.
Right. Okay, and I drew it that way. So these are first things that you need to realize is what goes with what. And when we're talking about why does A go with T and G go with C is when we have DNA, okay? And you'll see it drawn like this. You have a complement strand, you have the template and the complement, okay? So the template is usually what is red, and then you have a complement, which is exactly what it says. It's complement, the opposite part, okay? It makes it's a secondary because DNA is double helix. So if I were to draw this, A, T, C, G, A, T, C, G, A, T, C, G, and then we go in, and this is the template strand, and we go in, then I have to write its complement. And now we're still dealing with DNA, okay? So don't get confused with DNA and RNA and start thinking about bio here. We're just doing basic DNA. So its complement would be T, and then A, and then G and C. And then the same thing would go around. Okay, so this is just basic bio introduction with DNA and base pairing. So this is base pairing. You have one base and one base, and in between here is what we're going to start talking about is what is holding these together, okay? What type of bonding is occurring between these base pairs that holds them together? Yeah, okay, hydrogen bonding. Okay, and that's why most people believe that this is why that hydrogen bonding is so important. There are other forces, and you'll learn that for our sake right now, it's important to know that hydrogen bonding is taking place here. So as far as A's, and let's begin with A's and T's. So structurally speaking, you're going to have something that kind of looks like, I'm going to draw it this way. And then, actually, let's erase this. Make sure my carpets are right. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4. I need 1 more. 1, 2, 3, 4, 5, 6. Okay, so it's going to kind of look something like this. Okay, and this is adenine, right? So you don't, whatever you know the name, need to know the name or whatever. It doesn't matter. I'm just going to call this is the A base pair, one of the A's. Okay, So we decided that T is its base pair, and we know the type of bonding that's in between here, we decided is hydrogen bonding, right? So on this group, for A, we should have an O, okay? So if we look at this overall drawing of these base pairs, oh gosh. Right here, primarily, we're gonna have some sort of N, an O, and then we're gonna have another molecule here. You don't need to know exactly what this structure looks like. You just you don't need to know what, if it's an O or an N. You don't need to know what it is. Does that make sense? Like, you don't need to know this is carbon 2 and this is carbon 3. You don't need to know in detail. You just have to show that between this side of this A base, we have bonding occurring. So we have, this structure is going to come out like this. So we have these, actually undo. Okay, here, here, and then here. So these are the points. This is a chain, okay, coming off. This is a chain coming off. It is the red points that do the interaction, all right? And then I'll draw this interaction as yellow. So hydrogen bonding takes place here, here, and here. Now, these have to have carbon chains coming off, because if we just let everything attach here, there'd be some steric hindrance. So that's why this structure of A has these additional carbon chains, side chains coming out, because we have to allow for interactions to occur, right? We can't have everything kind of clumped in, and it just won't work. Okay, so, so here we have this, and then we said that its other base pair is T, so we'll draw that in green. And T, again, you don't necessarily need to know what these structures 100% look like. I would just draw them like this. I think it's 6, 2, 1, 2, 3, 4, 5, 6. Yeah, 1, 2, 1, 2. Yes. Yes. I'll show you, because these can also form. Let me get there. Okay. This bond here is a different type of bond. So Kennedy brought this up. This is a different type of bond. These two will be the H bonds. This is a salt bridge, okay? And we'll get in there. It's a minor groove. So just let me finish this one. Okay, so then we have these, and these H bonds look like this. Oh, we're getting kind of, it's really hard to draw this on here. I'm sorry. Okay, so we got, and they're gonna form here and here. Okay, and that's pretty much what happens, except for we have a little bit more structural. We have another carbon chain coming up here. So it looks like that. So carbon chain, hydrogen bonding, hydrogen bonding. This is another bridge though, because we do have this terminus here, and there is an interaction as well down here. Okay, and I'm just going to call this intermolecular. We don't need to know that as well, but you're gonna, if you look up structures of A's and T's, you're gonna see it built like this. So I just want you to be aware that there is another bond going on. So adenine and thymine form 2 hydrogen bonds. Okay, so that's important to know that we make note of that as it was made aware. So they form 2 bonds, 2 H bonds between A and T. Okay, and that's basically how it is. And then you can kind of draw the same, the structures look the exact same, six membered carbon. Okay, so between your G's and C's, it's going to look exactly like I just said, exactly the same. A 6-membered ring with a 6-membered ring, substituents, substituents, and they could be any substituents, and then we have bonding. The reason that we have this going on is just because of our pyramids and peridions. That goes into bigger detail of biochem, which I don't know if he's ever going to test you on that. So that's why we have 3 and 2, is just basically the size of these structures and the grooves, but you don't need to know that. Just note that these A's and T's form 2s, G's and C's have 3 H bonds. Okay? All right. So as far as number 1 goes, that's how you'll draw it. I would literally just draw out these 6-membered carbon rings and then draw substituents and then indicate these H bonds going out. Okay, so it doesn't matter. It doesn't really matter. You could put it, I would draw it like this though, with my H bonds here and then 3 H bonds here. We know this is H, H, H. Okay, that's the first question. Any questions on that question?
Should we draw so like, if you were to put like the GC one like underneath the AT, do you need to connect those to show that they're like the template strand and the complementary strand, or does it matter? Does that make sense?
Do we need to draw this thing?
Yeah, do we need to draw it like that? Like with
Nope. Okay. You don't need to draw that. I'm just showing that they were bridging. You can just leave them as A's over T's and T's over A's, just like that. Okay. Yep. So let's just draw it one more time clear for you guys, and I'll do it in different colors. Thymine, this, adenine, can I make this bigger? Oh, dang it. I ran out of room. Let me redraw that one. So we can see this. Those are the specific interactions. There's an N here. Okay, there's an actually, if I were to draw this more accurately, this N is not, it's not a carbon N. The N is actually here. Okay, it's built in here. So then you would have interaction here and an interaction here. And that's the 2 bonds. Because if you go back to thymine, you have another O here. Okay, so these are the 2 bonds, O to H. This is an H, not an N. You guys need to catch me on this stuff. You're drawing it down. This is an H2N. This is an N, NH, because this is going to make an H2N. This is going to make your OH. Still form of H bonding between the 2, total of 2 H bonds between A's and T's. Now, if we draw a clearer version of guanine, we'll have, they have another, just to let you know, there's more substituents out here, so I'll draw it like this, more things. But you guys don't really need to go into detail with that, just look at what's interacting, okay? So you could draw it like this on your paper, this little dash, this line out, and then a little wavy dash is indicative that there's more things on that group. Okay, so if you really want to be specific, you can do that. So then we'll have guanine, Cytosine. And built in there, it's not a carbon. And then we will have, are we starting to see how this works? Is this a little bit clearer now that I've redrawn it? And remember, this is going to have things down here, more substituents, and this is actually going to have a whole nother cyclic up here, so you can draw it like that. But this is where the interactions are happening. O to H, H to N. Remember, HN, any single interaction with an H and some electronegative atom is hydrogen bonding. And then again, H to O, and that's a double bond, we have hydrogen bonding. So that's a total of 3 H bonds.
Is the NH in the G one, is it in the
Yes, this
structure
is not
of
going to be a
the-
terminal. Yeah, this is not going to be a carbon. If they're on there like this one is built in. So that's an
Okay.
N built in. Yeah. One, two, three, 4, 5, 6. So right here, it's built in NH. Just like this one's built in. Built in, built in. This NH, built in. This built in. All of those are built in. That's not a carbon. So when you're actually drawing it, you should have it like this, right? Okay, so it's built in. So this is kind of, I think, a little bit clear between the interactions. So this is number 1, question number 1 right here. This is number 1. Let's see what else number 1's asking. Show how and they are recognized through hydrogen bonding. So these are Watson. I'm going to label it as BP, base pairs, with H bond. Okay, that's number 1. Screenshot it, take it, write it down. Let's move on. Now it says to draw the structure of 2 nucleotides, one from DNA and one from RNA. Draw out 2 nucleotides. Okay. How do you think? Well, how do we think we should begin with that? How would we start to draw out 2 nucleotides? That stinks that he's making you draw this out. Ugh, it's going to be a pain. Start with the sugar. All right, so what makes up a nucleotide? What is a nucleotide comprised of?
A phosphate, a sugar, and a base.
Right. These 3 things, that's what makes up a nucleotide. Cool. So let's go ahead and start drawing out just one of them. So if we're dealing with, remember, if we're dealing with DNA, right? We should see, and if we're dealing with RNA, what changes?
The T is a U.
The T becomes a U? Everyone agree? Or does the A become a U? Is it A or T that becomes a U?
T.
T?
T. Yes.
Yeah.
Okay. So we should have, this will stay. Yep. I just want to make sure people are aware. So the only thing that changes in DNA to RNA are these, is thymine. goes to uracil, okay? That's the only difference between the two. A lot of students always make it the A. I don't know why, but it's what they do. Okay, so now let's go and continue to drawing nucleotide. So for this one that I'm going to do is going to be in DNA, okay? And how do we differentiate? We'll talk about how we differentiate between DNA and RNA as far as nucleotides go. So let's start with what we'll call the triphosphate. Okay, so that just means tri meaning 3 and phosphate groups. Okay, so you'll have O, I should do this in a different color. We'll make it orange. Our phosphates will be orange. O, P, O, double bonded O, and another O minus. Okay, then we have this O attached to a P, O, another double bonded O, O minus. I should be drawing that the other way. That's really. Okay. Then we'll have one more. P, O, O, O minus. So 3 phosphates. That's your first group. Okay. This is the starting of our nucleotide. The next group that we're going to have is the sugar. Okay. So then this sugar is going to attach to this O here. So we'll have the sugar. And the sugar can be drawn, I'll draw it like this because we have this H group. And then it's this weird pentose shape, okay? So you would draw it like and remember, that's just indicative of where they lie on the plane, okay? These thick lines. And then we have an H here, OH here, H here, H here, and another back H down here, okay? I'll draw that line a little better. Okay. All right, so this is the sugar format, okay? This branch that comes up is what's gonna next connect the base a little bit. So, which leads us to our base. And in this case, it doesn't matter. I'm just gonna pick any base to draw, okay? So, but you'll be able to differentiate which one I am as we get further down. So then we'll have this will attach to an amine group. So we have N, and then we're going to start drawing our sugar or our base. So yikes. 5s are very hard to draw, by the way, so give me a minute. And then up here, I'm going to have to erase this really fast. Goodbye. Okay. This right here is a nucleotide. Okay, this is how you'll draw the nucleotide. You have triphosphate, a sugar, and then you have a base. Okay, so which is basically this, the reason that and then so this is your source of your nitrogen, and this is your source of your oxygen. Okay, and that's why it's important to understand what these look like, because that's where things will interact. Okay, the sugar is just the middleman that bridges the two together. Cool. So that right there should answer draw full structure. 2 nucleotides, you literally can just repeat. Now, one of them has to be in DNA. So how do we differentiate the sugars between DNA and RNA? Do we know? Yes, OH group. So the one that I drew, which one is this? Which one is this? What sugar is this?
DNA.
This is DNA? This one's DNA? Is that what I heard? Okay, why? Because of this group right here. And now let's see if I can pick a different color. This one. And right here, okay? This is an H, so this is deoxyribose. Deoxy, what is that? All you need to know about deoxy without oxygen. So deoxyribonucleic acid, okay? And so if I were, can I erase this? Can I erase this? We good? Okay. If I were to draw, I'll just do this all in black. If I were to draw ribose, okay, so that was deoxyribose. This would be ribose. Remember, this is an interaction over somewhere yonder, okay? So look at this one, he changed. Okay? So this versus this. These are your big key identifiers. Okay? So when you're going to draw your second nucleic acid, I'm not going to obviously do your entire lab for you. I'll do 90% of it. You're going to do the same bases. So start with your phosphate, switch out your sugars, draw a base. Okay? It didn't ask to me, it didn't say to specify the types of bases, but what I would say is if you wanted to be an overachiever, you could draw on the RNA nucleotide, you could use a uracil as a base and just label it uracil, or look up uracil and kind of just draw that, mimic that for your base. And then you will change out the sugars. And then when it asks for point out the key differences, you could say RNA is made up of ribose sugar, DNA is made up of deoxyribose sugar, and those are found on these particular carbons, right? Which carbons? And also the bases have an opportunity to change with regards to thymine, right? So we have T in DNA, but we have U in RNA, and that will be sufficient for answering question 2, okay? Any questions, comments, concerns on that? Did we all get those structures? Do you want me to draw again? Yes. When it's asking for the full thing, you have to draw both. He says to draw 2 structures, so just to be safe, I would draw the whole new triphosphate group, the new sugar, and then I would draw the new base. Make this U for RNA, uracil, and then I would make this T in DNA. And then you'll swap out these, but this has to stay. Cool. All right. Question number 3. Write out an exam. Oh, somebody was trying to get in. Oh my gosh, I wonder how long they've been waiting. Sorry. Yes, that is the key difference between the sugars. Yep, and the base pairs regarding A's or T's and U's. And then the sugar always bonds with the O from the triphosphate, correct, no matter the sugar. Yep, because it's actually interacting with, when I draw the sugar out, right? Because so this is the only group that changes here. This can be that, or it could be this. This one's not actually doing the interaction. This is always doing the interaction, this group up here, and this one never changes when you attach to the phosphate. Okay. pH on phosphates. So it doesn't matter. This interaction will always occur. No change. This is the only thing that changes. And really, it's just used for identification purposes when we're talking about where our DNA or RNA or where these base pairs or where these nucleotides are coming from. That's how we can differentiate. Okay. Write down an example of a condensation reaction forming a phosphate sugar bond. All right, let's go ahead and write that question out. Okay. Yikes. Okay. Write out now that we know a little bit more about our nucleotides, what is this asking? Let's look at the key identifying words in this statement. What is it that this question is specifically asking us to do? What's the terminology? First of all, what's a condensation reaction?
You add like water to the reaction. It's like hydrolysis, maybe.
So what is it? So does it add water or does it?
It's
producing. It produces water. Okay. So are we adding water to the equation?
No.
So where should water be when we write this out? Left or right side?
Right.
Right.
Right side. Cool. So the first things first is that I want to make sure nobody's asking. You need to know this word, condensation, to mean to produce water. So we know we're going to be producing H2O. So for that, it has to be on the PDT product side. Okay, that's how I abbreviate product. Cool. So here's what I'm going to draw. You're going to make something called a phosphodiester bond. Okay, phosphodiester bond is what interacts between the hydroxyl group on the phosphate going to the sugar. So that's the phosphate sugar. It's called a phosphodiester bond. Please know that word. You will probably put that on an exam and ask you what is happening. So a bond between the phosphate group and the sugar is a phosphodiester bond, okay, of nucleotide. So let's just say, hypothetically speaking, I don't know how he wants this. He want me to actually draw it as an equation? Probably. Oh, no, it says write out an example. Okay, so we don't actually have to do, okay, cool. So what we can draw then is this. I like structures because that's the organic chemist in me. So let's draw more structures. I'm going to erase this because I need more room. So clear. Let's do it in purple because it's fun. All right, so here we have one base. Okay, so let's go ahead and this is happening between the bases. Well, we have some base. I'm going to condense the structure. I'm not drawing 3 phosphates anymore, okay, because it's too much for me to draw. But you have to just assume that this is happening with one of them. So we have one phosphate group, right? And then we're going to start going to our sugar. So that's going to look like this. 1, 2, 3, 4. Oh crap. It's so hard to draw this. I'll put a C here and then I'll do it like this. Okay, so then here we'll have OH. And here we'll have an OH. So what sugar is this? What sugar is this?
Ribose.
Ribose. Remember, deoxyribose. This is the key spot. This is 2 OHs is ribose. Cool. Did I hear you correctly? A bond between the phosphate? Yes. Bond between the phosphate and the sugar is a phosphodiester bond. This is ribose. So where would we find ribose?
RNA.
Perfect. Let's start. Every single time we draw out, yes, sugar's always 5. Every time we draw, it's a pentose, right? Every time that we draw these structures, I want you to keep asking, or you're looking at structures, I want you to start looking at specific components, identifying the phosphate group, identifying the sugar group, identifying which sugar group it is. As far as the bases go, I'm not going to draw the base out, guys. I'm just going to do this, and I'm going to write base. Why? Because this really doesn't matter right now. Okay, so that's how it's going to look structurally. Okay, but that you can go and kind of determine if you want to take more chemistry. You could differentiate those bases, but for right now, not pertinent. We're looking at just the bond interaction. So I have a base, and so this is one nucleotide. Okay, this is all a nucleotide. And then let's say I have another phosphate group from another nucleotide, and I have this over here. So it's going to look like this. Let's do a different color, friends. So you have to have this, you have to have 2 nucleotides for this to work. So we'll draw another one like this. But this is going to have an OH group. What does that mean that happened to that phosphate? What happened to it? Because remember, when we drew these phosphates, this triphosphate, every single one of them was an O minus, and a double bonded O. So we would have had O minus here, but this one usually is the bridge that connects, like this right here was the bridge that connects the sugar, this O. So what happened here to have an H on it? What kind of interaction? Yeah, it was definitely protonated. So it could have had its phosphate cut off, right? And so then it would be diphosphate. It could have gone through any sort of reactivity or connected again to, because we have ATP and then we have ADP, right? Adenine tri, adenine di. So it could have gone through a plethora of things to at least get protonated of some sort. But that's going to really help us when it talks about with regards to how these bonds are interacting. Okay, so that is the source. This will become the source for how these two things react. So if I have this, where is this going to interact? Now remember, we are going to be losing water, right? Well, we're going to be losing water from this. These are our reactants. We're going to be losing water from this. So out of here, where do you think this OH is going to attach the most? Remembering the structure of an amino acid. Again, I'll rewrite this out. We've got phosphate to sugar to base. Phosphate, sugar, base. Phosphate, sugar, base, phosphate, sugar, base. So where do you think this interaction is going to occur? Is this OH going to work with a phosphate group, or is it going to work with the sugar group?
Sugar.
Okay, where is it going to react?
The OH on the
Which OH?
third
This
part?
OH on the 3. So is it this one, or is it this one? This one?
Yeah,
Okay, perfect. Why?
because 5' to 3'.
Correct. Okay. 5', 3' building is only going, So that's why we only look at these two. The one on the right is identifying if it's which type of sugar. The left is the interactive sort. So depending on, just remember that this particular sugar, this particular OH group is how we will join, okay? This is how we'll join it. This will identify what it is, what type of sugar. This is the source of reactivity, okay? So in order for that to occur, oh, do I have a chat? No, yeah, okay. So in order, now I'm gonna have, is everyone good? Can I erase this? We good? So then to draw the full complete structure, okay, remember you have those two nucleotides, the full structure will then turn into this. That's a 2. Okay, and then remember, this is still staying here because we're in ribose, okay? This was our reactivity spot. So that will become this. And if you go back to your notes, which I know everybody was drawing, we'll see that you know what we'll see that is you have to draw it this way moving forward. This H has to get removed. So then we'll be the conjoining of our new phosphate. And then I'm not gonna draw it all, but I'll do this. Okay, and then that's where the sugar will be. Cool. So this is where the phosphate will attach on this 5' on this carbon, this carbonyl with this O. Okay, this is your source. So this is how you'll answer the question. Draw a condensation. Also, you need to add this to it. Condensation, lose water. Where do these go? Overall, this is a phospho. And this phosphodiester bond took place here. Okay, this is the interactive site. When we draw this for the lab, are we supposed to draw triphosphate, or is it just you don't have to draw out those 3 sugars. I would literally just draw it like what I drew here. Just a simple phosphate, because you know it's annoying to draw all those out. You just need to indicate that that's what's happening. It's between a phosphate group and then the location of the OH on the sugar, and that's where that other phosphate will attach. Okay, so I would just draw it like this, and I think that would be totally acceptable. I think it becomes redundant. You already drew it 3 times on the previous question. Now we need to write out an example of a condensation reaction between a nucleic acid base and sugar. Okay, any questions on phosphodiester bonds? And the 3' on the first sugar, is there an H above the O? Yeah. Because this is a carbon. Carbon forms 4, so 1, 2. Carbon to O is 3, H is 4. Yep. I usually don't draw in hydrogen, so if you would like me to do that moving forward, just let me know, okay? Cool. Any questions on this? So the bonding, the joining, again, to reiterate, the condensation reactions in nucleotides can occur between the phosphate and the sugar, and we call those phosphodiester, and then they form a phosphodiester bond. That's the bridge that you need to take away. This is the takeaway message. So, and when I say lose H2O, you have to think about from the products, we're losing water, right? We're losing water. If I were to say we're gaining water, it technically would still be the same thing because we are gaining water in the product side, but from that bond alone, we lose the water. The water is lost. We get it out of that system, okay? That's to condensate. Cool. All right. Let's go ahead and I got too many things going on open here. Hold on just 1 second, okay? And my little kid is over here. Nope, things are freezing. All right, let's continue. Now we're going to do react. Okay, so now this one is going to be still condensation, but between the nucleic acid, base, now how the heck would we draw this? How would we even start this? So now we're dealing with nucleic acids, the base, so condensation. Between the base and the sugar of a nucleotide. So now that we've just learned about a phosphodiester, which is the phosphate and the sugar, how can we take that sugar and connect it to the base? We need to think about what type of bond that is. So what do you think, how would I do that? Where do you think I should start with that? On the sugar. Okay, so let's start with the sugar. So I'm not going to draw phosphates now. We good? I'm not going to draw any phosphates because we're not in dealing with the phosphate. So let's go ahead and draw the sugar. Something that looks like this, remember. We're still dealing with ribose. I like ribose. So now, where on this sugar, I have some sort of base up here, right? That this is connected to, because if you look at the previous structures of our nucleic acids that I drew, that is where, that's where most of it, okay, so what kind of reaction is this then? If I have an N based pair on here for my base, if my base has an N to it, what kind of bonding is happening here?
Hydrogen.
Okay, so there's hydrogen bonding. And what else? So we're saying that it's a condensation reaction. No thoughts? Yeah, it'll be that. I'm trying to think though, if we're having a condensation reaction between a base and a sugar, then we're still getting what? What's leaving the system?
Water.
Okay, so how between a base and here can I draw this occurring? How would that happen on a base? We can do it. We can do it. We can do it.
Would there be an OH group on like the top of the line by the base?
Okay, so what kind of base do you want me to draw? Let's get an example. Do you guys have notes open and things like that? Do we have bases?
Can you do a cytosine?
Right. So let's see. Let's clear this. Okay, cool. Okay. Yeah. So I want you to start looking at some bases. There's only 4 of them. I know it's hard to asking you guys to memorize these things, but it's really nice to kind of see how these will bridge. So if I have this sugar, which we've already determined what it is, okay? And, oh man, come on. Right, and I have the OHs here. And what if I had this carbon and then I had some OH on this? Is this possible? This is a carbon here. Can I have this?
Yes.
Yeah, yeah, okay. Where would this come from on this sugar? Is it on this sugar?
Yeah.
Okay, but have you seen a sugar, have we drawn out ribose or deoxyribose with an OH group? That's deoxyribose. What's this, for this OH to occur, it could be on deoxyribose. So deoxyribose looks like this. We have an OH group right here. Oh, it's right here. So in a condensation reaction between a base and a sugar, you want to use deoxyribose. The last one we used ribose. So ribose sugars have phosphodiester bonds. So phosphodiester would be between the phosphate and the 5' OH group, right? And then we have, which was right here. But on deoxyribose, the condensation reaction will happen between the base and the sugar. Are we kind of connecting bridges here?
Yes.
So condensation reactions in ribose sugars, aka RNA, form phosphodiester bonds. Okay, this condensation reaction that occurs between deoxyribose sugar and a base will occur, like I said, in deoxyribose. So then we'll have this OH group, and then the base that we have, which can be any base, will have some sort of NH. Okay, so if I had this and I drew it like, you know, this, some ugly tense thing, just looks like this. I have an N here. Okay, this is where you'll have it. And again, we're going to be losing this H to OH component is what's lost. And that's how we see all of our sugars or all of the bases with that N drawn in. There. Because the only thing that connected this was the original OH, but that OH was lost via a condensation reaction just to attach that N from a base, a nitrogenous base. Remember, we need to start thinking that bases are full of amines, right? That's why they're called amino acids. They have a lot of nitro, they're nitrogenous, okay? So, you know, you are gonna have some sort of, so you can't have phosphodiester bonds in deoxyribose. You can have it. It's not just limiting, because phosphodiester is just between the bond force between a phosphate and a sugar. So you can have it in any sort of nucleotide. I should have clarified, phosphodiester bonds can still occur, but I'm just saying when we drew focusing on the phosphodiester bond at the last example, that was a ribose, okay? And so, you know, I just want to, because yeah, up here you'll still have, this is the phosphate group, and so you can still have phosphodiester bonds that form here, right? Because this is the source of reactivity, whether you're deoxyribose or ribose. So phosphodiester bonds form here, and then you have bond loss of condensation reaction between the base and some sugar group. So your sugars should always be just like this. all your sugars should be drawn. Either they should all have this. This is what they all should have, and they'll draw this in green. You're always gonna have this. It's always gonna be 5. You're always gonna have this, and you're always gonna have this, and you're always gonna have, I'll draw it like this, P. Okay. So you always have this. All your base sugars should look like this. How do we differentiate? I will draw in purple. This changes. I'm going to put an X here. This will change. Okay. This will always occur. This will always occur. You'll always have some sort of phosphodiester bond via condensation, and you always have to attach some sort of base on this OH via condensation. Okay, and that's how we create that nitrogenous base. And there's the phosphor. So this is the only thing that changes. Source of reactivity, source of reactivity. We'll draw those in blue. Reactivity, reactivity. Identify. Identification of sugar. Are we good? Clear. Whether you're on a ribose or a deoxyribose, 2 sources of reactivity sites are these OH groups. Okay, that's where things will happen. This one will attach the base. This one will attach the phosphate. Okay. Any questions? So for that question, you can literally just draw this up here. You don't even need to include the phosphate. You can draw this sugar. You can draw it as either deoxy, or you could do it as ribose. Either way, because this is not going to change. You have to draw this OH group and then draw a bridge forming between some sort of base, some N component, some amine component of a base. Okay? That's going to get lost. This is what's going to get lost. So then make sure you add plus water. Cool? And then
we don't
question.
sorry.
Well, go ahead.
So we don't have to write the green arrow to the red sugar. That's sugar.
No, here, I'll draw it nicer for you guys because I know people like to see that. I would just draw it like this. I'm putting this here because you can choose either one you want. Okay, so this honestly is the base. Okay. And I'm going to draw it like this. It'll have an NH, okay? Because then this yields this plus. I put this N here because that's what replaced the OH originally. And then I'm writing base because you can choose to draw anything you want there. And that's the phosphate group.
So then, Liz, that last thing that
Yeah.
you're drawing, so the H2O plus that bottom part that you just finished, that's the nucleotide.
Yes. This is the new.
Okay, so it's H2O plus the full complete nucleotide.
H2O plus the combination of these two.
Perfect. Okay.
I'll put a P for phosphate here. because you have some sort of phosphate bond. Yep. And then we're adding the base. So that's how you'll connect a base to a sugar via condensation. And that's how I would draw it. I don't draw things elaborately because if it's not interacting, why do we need to draw it out? I just feel like that's just only practice for you. Just indicate that there's still a phosphate group, right? We didn't lose anything there, but I just don't feel the need. I do draw that. This one is nice to draw, but like a full molecule, I mean, I don't see him asking for that. It just wants between the phosphate bonds, so that's why I only drew the amine that way. But like the whole, like you don't need to draw something that looks like this. I think it's just asking too much. But there's people on here that will email me, so I will. Like this is an entire amino acid here. And I just think it becomes a little bit redundant to do it this way, but okay, so this is the source, this is an NH, this is the source from the O, H of the sugar, okay? So like this is technically a full base, but see how it's just a lot to draw. So I don't really think it's necessary to do that. Cool. Sorry, I'm getting a familial call right now. Okay, and state the central dogma of biochemistry. There you go. That's it. Any questions on the central dogma? I'm sure you guys have all heard of the central dogma, right? Because without DNA, we can't build RNA, and RNA is what makes proteins. Proteins are made up of what?
Amino acids.
Yes, I would hope that we're kind of clicking here. What are carbohydrates made up of? What are their building blocks? Monosaccharides, right? And what are lipids and fats made up of? What are their building blocks? Nobody knows. I find that very hard to believe, that nobody knows what are the fundamental building blocks. Sugars, lipids and fats, or sugars, carbohydrates? Yeah. Cool. Okay, so when we're talking about the central dogma, you have to understand there's building blocks. I call them building blocks, okay? There's main big ones. These are the big ones, right? These are the little blocks. That's what I'm talking about, and he'll use this terminology. Big blocks, these are the little blocks. Nucleic acids. Nucleic acids make up nucleotides. Amino acids make up proteins. Monosaccharides make up carbohydrates. Lipids and fats are full of phospholipids. Okay, should we identify replication, transcription, translation? Let me go through his exams. That's really biology related. I do know from past exams, you will need to know the structures of your proteins, and you will need to know alpha, beta, pleated sheets, primary, secondary, tertiary, quaternary stages. You will need to know, what other notes do I make? You will need to know enzymes, which we should already know plenty about, but I'm talking about like induced fit, lock and key, knowing the polarity or the acidic or basic components of amino acids, and reversible inhibitors. And again, that's all stemming from enzymes. So enzymes, amino acids, right? Because what are enzymes? Those are proteins. So you need to know about enzymes, okay? As far as transcription translation, I have not seen that on his exam. So I wouldn't be too concerned with knowing the specificity of that. I can kind of give you a tiny glimpse of it if you'd like, but I don't think you need to know that, because replication is more genetics. That's all with genetics, and you don't need to know that. He just wants you to know about DNA, RNA base pairs, because those base pairs involve hydrogen bonding, and then that's the biggest component why we introduce DNA, RNA into this class. It's not necessarily more so of the genetic side, it's just because we can talk about hydrogen bonding. Okay? Any other questions, comments, concerns about this nucleic acid lab, amino acids, anything of the sort? If I were you, again, to reiterate, please, please, please at least memorize or have easily accessible your acidic and basic amino acids. If you know those, then you can differentiate everything else. You don't even need to know the names of the other 15 amino acids. Just know your acidic ones and your basics. There's 3 and 2. Just know those 5, and then the rest of them doesn't even matter. It doesn't even matter. So if he asks you about phenylalanine, I know it's not acidic or basic, so it's got to be nonpolar, right? Nonpolar covalent. Things like that. Again, salt bridges are important to know what happens between a salt bridge. I showed you that. And those occur in proteins. Okay, a lot of students start blending amino acids and nucleic acids. Okay, amino acids are proteins. Nucleic acids are nucleotides, which is specific to DNA. Okay, even though the central dogma is DNA, RNA, protein, remember proteins come after. You have to start with the nucleotides and the nucleic acids. You have to start there, so that you can create proteins and things of that sort. Okay. Could you explain number 2 on the amino acid lab? There's a number of different tests for amino, and choose one and just, okay. Yeah. Okay. Hold on just a second. Okay, let's see. So you want to know, like, all right, I get you. So we can do like Comase blue and things like that, I think are one of them. Okay. So for that question, what did you have written down? Did you have anything? Does anybody would like to share what kind of test they used to determine proteins and amino acids?
I did the ninhydrin test.
That's a good one.
It's purple, and it's pretty easy to explain if you want to do that one.
Yep. So. Okay. What else? This is important to know that it tests for a free NH2 group. That's what that tests for. And then if it does, so when this group is present, that's when you'll get your purple blue. If you use this, there you go. If you use this test though, okay, and say you have, and some sort of nitric acid is present, you'll have presence of tryptophan. So really it's nice to know if we're looking for tryptophan. That's why a lot of people use an inhibin test, because of this guy. They like to test it. In that case, it'll turn yellow. So it just depends on what it is that you're, I should learn how to spell tryptophan. So it just depends on what it is that you're testing for, but really you're testing for this nitrogenous base here, this NH2, and it has to be free. And so, yeah, you could use that one. What other ones do other people use? It'll be yellow. Sometimes people also see this as like brown, but really it should look like this. What other tests have you guys used? There's more. Millions? Okay, what did millions, the millions test for? So this test, the Bure test, yep, this will test for specifically if tyrosine is present, okay? The Bure is just a general test. So you can go and you can say, okay, what if this test will just test for general proteins? Then this too will create a purple, right? And it'll react with copper sulfate. So you can talk about the Bure test, an inhibin test. Ones that we technically do are the Bure or the inhibin. So you can do either one of these. When we did our lab, we used both of those. Okay, we didn't use the millions so much. Oh, dang it! But you could do either of those options. Any other questions? Okay, in the chat, yep, good. In the chat, I want to do a review session. I'd like to do one this weekend. This week is really crappy. I have a lot of things going on because remember, I'm still a student too. So I would like to hold a review session, and in the review session, I'm gonna strictly go over what will be on the first part of your last exam and then work on the cumulative review. So I would like to do, hold one this weekend. What time and dates work best for people on the weekend? Sundays, do those work? Does Sunday work? I don't like to do Saturdays because we're just coming off of working school on Friday. And so I don't really like to, I should stop sharing this because it's not pertinent anymore. I don't really like doing things on Saturday. I feel like we need to have a break, but on Sunday, okay, so let's say Sunday, times for Sunday. We could do early, like 10, 11. Everyone really likes 11. So a lot of people also like 1 o'clock. So what I could do is just kind of have a running Zoom session from 11 to like 2. And then that way, if you want to come for the first, I'll just keep repeating the information. But I could do like 11 to 1 or 11 to 2. And then that way you guys can pop in at 1 o'clock. I don't think going later in the evening is nice because we'll do 11 to 2 is great. Okay, so this Sunday, cool. So this Sunday, I'll after this meeting, after the Zoom, I'm gonna close out this Zoom session. So after today, just keep finishing, submit your nucleic acid amino tests or your labs, get those submitted. If you want to resend me what you have drawn out and done, I can double check it before you submit it, but just get these labs out of the way, okay? Get these labs out of the way so that we can focus on the remainder of the semester. I did want to talk to you guys about the capstone, I think it's My Life, and how that works, because I've had some emails about that as well. Please know that